We are live on YouTube, Juani.
Okay, so can you start right now?
Yeah, you can start.
Okay, so good afternoon, everyone. It's a pleasure to be here in the BSB conference. We will start the second session from BSB conference. We have for this session five papers, five full papers. Most of them are related to some way to data analytics on biological data sets. Like it could be either tabular data sets or either image, but it's really interesting. I'm really excited and honored to be here chairing this session. So for everyone that to be presenting this afternoon, we have 20 minutes. So please pay attention to the time that you have. And we will have at least, let's say, 10 minutes for questions and discussion by the end of each one presentation. So another point that I'd like to mention here is that I will let you know, when we be, running out the time. So pay attention to these, and I hope that everyone enjoyed this session. And I would like to start our session with the paper presented by, I think that to be Gabriel, and the paper is COVID-19 X-ray image diagnostics with deep neural network. So Gabriel Oliveira, please start.
I'm going to share my screen.
Okay.
Can I start?
Yes, please.
Hi, my name is Gabriel Oliveira. The title of this work is COVID-19 X-ray image diagnostic with deep neural networks. The authors of this work are myself, Rafael Padilha, André Dorte, Luiz Cereda, Luiz Miyazaki, Maurício Lopes, and Zanoni Dias. This is the agenda of the presentation. The outbreak of COVID-19 has impacted the public health and economy of many countries around the world. Many researchers have been made to trace the origin of the disease, to evaluate treatment methods, and to find efficient diagnostic methods to prevent the disease spread. With the recent success of machine learning methods applied to image analysis, recent works in the literature use this technique to perform COVID-19 diagnostics, mainly using convolutional neural networks to classify chest X-ray images or computed tomography scans. Training a convolutional neural network from scratch is challenging because the optimization process requires a lot of labeled data, which are not always available for medical problems. With that, transfer learning and fine-tuning techniques can be used in the convolutional neural networks for medical tasks, such as COVID-19 diagnostics. In this work, we perform an extensive evaluation of convolutional neural networks architectures published in the recent years in the literature for predicting COVID-19 pneumonia and healthy patients using chest X-ray images. We use transfer learning techniques to adapt the convolutional neural networks for this domain, comparing them with traditional machine learning classifiers. To evaluate our method, we employ the COVID-X dataset, which has images from several different datasets. The images available in this database are divided into three different classes: COVID-19, pneumonia, and normal, which is a patient with no disease. The table presents the distribution of patients and chest radiography images in the train and test sets. The figure illustrates examples of normal, pneumonia, and COVID-19 chest X-ray images. Some X-ray images in the dataset present artifacts and noise patterns such as medical devices connected to the patient, contour and volume of the breasts, difference in size, and shape of the rib cage due to the patient being an adult or a child. Even though these patterns might be common to the task, they are not directly related to diagnostic outcome, and the model should learn how to ignore them. Our method is divided into data preparation, model selection, and classification. We consider two different scenarios for the classification. In the first scenario, we apply the model for multi-class classification with three possible outcomes: COVID-19, pneumonia, and normal. In the second scenario, we consider a binary classification of COVID-19 versus non-COVID, grouping normal and pneumonia as non-COVID class. During the training step, we resize each image to the network expected input size and normalize each pixel values. Also, we employ data augmentation techniques to increase our training set and add more variability to the images. We apply rotations, zoom, vertical and horizontal shifts, generating 10,000 virtual images for each image in the training set. We evaluated several convolutional neural networks proposed in the past 5 years in the literature. For each network, we initialized it with weights pre-trained on the ImageNet dataset and fine-tuned it for the COVID-19 diagnosis. We adapted the network endpoint by exchanging the original output layer for a new output layer with 3 or 2 neurons, depending if we are considering the multi-class or binary classification. In comparison, we used traditional machine learning classifiers as baselines for this task. As this classifier received two-dimensional array as input, we serialize each image before feeding to each classifier. As each neural network has different learning process, the final characteristics learned by each one can be complementary. So we investigate ensemble of the networks to improve the results. We explore 3 different approaches of fusion. The first one, we average the answers of all convolutional neural networks, making the mean consensus between them. The second approach that we used was using the average, the responsing, and train a meta-classifier using the output probabilities of each network. The last method that we used, we extract features from the penultimate layer of each network and train a meta-classifier with this information. As meta-classifiers, we apply support vector machines, random forests, and shallow neural networks. To train the models, we divided the original training set into train and validation. First, we used 473 X-ray images of each class divided into 383 images for training and 90 images for validation without data augmentation. This was done to mitigate the class imbalance of the dataset, making a balanced subsample that allows us to train each architecture efficiently. Using this reduced balanced set, we evaluated all methods in terms of balanced accuracy and number of parameters for the multi-class classification scenario. In the first evaluation, we trained support vector machines with linear, polynomial, and RBF kernels, random forests, logistic regressions, and XGBoost classifiers with hyperparameters found through a grid search on the balanced set. For the convolutional neural networks, we used transfer learning and update only the new edge classification layers specific to our task. The table presents the results of the evaluated methods on the balanced dataset. The results on the validation set shows that the traditional machine learning classifiers can achieve better results compared to some convolutional neural networks architectures. The main drawback of the traditional methods is that the models lose spatial structures of images, which hinders their performance. The convolutional neural networks that were outperformed by the most baselines have an increased number of parameters, which indicates that the network size does not are directly related to the accuracy in the task. Our experiments show that the ResNet 50 architecture achieved the highest accuracy in the validation set, with an intermediate number of parameters among the networks evaluated. We select ResNet 50, EfficientNet B7, MobileNet V2, and Random Forests as the base models to perform additional explorations. We evaluate the three top architectures in the complete unbalanced dataset. We split the complete dataset, the complete training set of the complete dataset into 80% for training and 20% for validation in a stratified division, and we assigned weights to each class according to their size. Besides the classification, we employed the data augmentation techniques to increase the diversity in our training. Consider the increased amount of data. We unfroze the initial and intermediate layers of each model, allowing the update of their weights. We consider the two classification scenarios. The convolutional neural networks obtained high accuracy with ResNet 50 and MobileNet V2 outperforming EfficientNet B7 in both classification scenarios. All the networks overcome the random forest classifier because this classifier cannot deal with the overfit and difficult with the balanced dataset. After, we investigate the fusion of networks with average consensus and meta-classifiers using the probabilities. The first line presents the results using the average consensus of the classifiers. The remaining lines show the fusion using meta-classifiers. The choice of the hyperparameters was found on the validation set. The results show that the ensemble using meta-classifiers is better than the average consensus. Also, the fusion using meta-classifiers improved the results compared to the networks alone. Finally, we applied the deep features to make the fusion using meta-classifiers. The results achieved were worse than the fusion methods using probabilities and the networks alone. In the end, ResNet 50 was the best convolutional neural networks at both scenarios, and the overall best result was obtained with shallow neural network as metaclassifier using probabilities, achieving 92% of balanced accuracy on the multi-class task and 93.5% of balanced accuracy on the binary classification. We compare the confusion matrix between ResNet 50 network and the ensemble using shallow networks on top of probabilities of the ResNet 50, MobileNet V2, and EfficientNet B7. In our case, the methods are based towards classifying COVID-19 samples as normal or pneumonia, presenting the majority of the false predictions in the bottom row of each confusion matrix. Also, the ensemble approach is able to correctly classify 89% of COVID X-rays. As conclusions, transfer learning techniques can achieve good results by leveraging the generalization of the initial layers in a different domain. Also, ensemble techniques improved the results compared to the standard networks, obtaining 92% of balanced accuracy in the test set of the multi-class classification and 93.5% of balanced accuracy in the binary classification. As future work, pre-processing steps such as segmentation of the image, apply filters to reduce the noise, and use images with high resolution can be investigated. In the post-processing steps, explainability techniques can be used to interpret the results. Also, a continuous data collection is essential for future research in this problem. Thank you.
Okay, thank you, Gabriel, for your presentation. So we have plenty of time for questions. Let's see what we have. I don't know if people, I mean, you can put your questions in the chat, or either you can talk, I mean, if you're able to open your microphone. So if you don't have any question, I can make some. Okay, no question. Okay, so again, thank you for your presentation, Gabriel. I think it was really interesting, and I think that you, I mean, pay attention to all the, how can I say, steps in machine learning, let's say, to build your models. And I'd like to ask you, what was, I mean, your, how can I say, it was the main reason for choosing that architecture is, I mean, the CNN architecture that you selected. I mean, it was like, if I'm not wrong, I mean, you select 18 CNN architectures, and I mean, they are pretty different. I mean, they are pretty different the way they build them. I mean, all the layers and all the stuff, all the pooling, all the convolution. And it's really interesting because you can see that, I mean, by the time that when you present, I mean, all the parameters that they estimate, I mean, for each one, I mean, to have the model in the end. And you can see that there are some of them that they estimate like 25 million of parameters, and on the other hand, there are some that present only 3 million of parameters. So it's really interesting to see that sometimes, I mean, we can achieve, I mean, really nice, I mean, results with, I mean, a soft, let's say, soft CNN. And my question first is, has to do about, I mean, what was the main reason to choose this H architecture? For instance, we know that there are a lot of other, how can I say, well-known architecture like AlexNet, for instance. And I mean, what was your main motivation for choosing these CNN architectures? And I was curious about how long does it take to build up all this stuff, to build all these results?
About the first question, we selected the top architectures that were presented in the literature the past 5 years. AlexNet was architecture that was presented less than 5 years, so we didn't select this architecture. About the second question is about, I don't remember the second question.
Yep. The second question was about the time. I mean, how long does it take to have all, I mean, these? Because, I mean, if I understand correctly, I mean, you put all these architectures, I mean, all the CNN models, and then you realize that you come up with one, if I'm not wrong, Mobile 2. Mobile 2 has a really pretty nice result. And I mean, I think that it took lots of time. I mean, and I was just curious, I mean, about how long does it take, I mean, to have all of these models, I mean, using the CNN.
In the first experiment, we only trained the new layers put in the end of the architecture. So was really, really fast. In the second experiment that we used the whole data set, we trained all the architecture. So we unfroze the layers, and it takes close to 5 hours per network.
Okay. I don't know if anyone has some question. Let's check again the chat. No one. I can make you the last one. I mean, about, I mean, what was the criteria? I mean, because usually these, how can I say, image detection problems, people, they take the data set like in splits of training, validation, and test. In your case, I mean, you only do that in training test. And it was just, I mean, interesting to hear from you. I mean, what was the criterion to select, I mean, to use this kind of methodology instead of using the classical, I mean, train, test, and validation. So let's say that you have a 70% for training, 20 for, I mean, test, and 10 for validation. So to make sure that we really have, I mean, in the end, an independent test for checking your model views.
We selected the 80% for training and 20% for validation because the data set has only the train and test set. So we need to create a validation set. And this percentage of the training and validation from the original training set, we used the standard percentage of the split. So we used the 80% for training and 20 for validation.
Okay. Okay. Thank you very much for your presentation. I don't have any other question. I don't know if anyone has, I mean, any other question in the chat. No. So far, no questions. So we can. I thank you again, Gabriel, for your presentation. It's really nice paper. I was, I mean, before we finish your presentation, I think that there are some interesting aspects about, I mean, having more data sets, because as you said, I mean, you use some data augmentation, I mean, in order to improve your image. And I don't know if you're familiar with the GAN, so the adversarial networks. Have you heard about this? Maybe you can have the chance, I mean, to generate more image from the image that you already have. And I don't know, maybe this can help you to improve your model. So my last, I mean, my last point. But I really enjoyed the presentation. Thank you very much.
Thank you.
Okay, so we can go to the next, I mean, presentation. So I think that the Guilherme Sato, and he presents to us, I mean, the paper entitled Classification of Musculoskeletal Abnormality with the CNN, so Convolutional Neural Network. Okay, Guilherme, please.
Thank you, Ronnie. Okay, can you see my screen?
Yes, you can start.
Okay. Oh, hello everyone. My name is Guilherme, and today I'll be presenting our work on classification of musculoskeletal abnormalities with convolutional neural networks. So our motivation to work on this problem is that musculoskeletal abnormalities are very common in the population, affecting over 1.3 billion people worldwide. But diagnosing it is a complex task, which requires highly trained medical staff. And if someone is unable to obtain a correct diagnosis, they are unable to go under treatment, or maybe they will receive an incorrect treatment, which may lead to further complications such as chronic pain. So to formalize our problem, we are diagnosing abnormalities from radiographs of the upper limb, which includes the shoulder, forearm, wrist, humerus, elbow, hand, and finger. Now, while we could predict the body part in the image, it is hard to draw the line as where, for example, an image stops being a finger X-ray and becomes a hand X-ray. So our goal is to simply output whether an X-ray is normal or not, making it a binary classification problem. So to approach this problem, we apply machine learning, computer vision, and convolutional neural networks. These techniques have produced state-of-the-art results in other applications, including other computer vision problems, showing promising results in a wide range of problems. So naturally, to train this machine learning classifier, we need data, and we got this data from the Mura dataset. This dataset includes about 40,000 images labeled by radiologists, and each image labeled by the body part it represents and the normal or abnormal diagnosis. The images are divided into so-called studies, and here we have an example of a study composed of 4 radiographs. It's all of the same body part of the same patient, but from different angles. And to measure the performance of our classifier, we split the data into 3 sets for training, validation, and test. The overall approach we use here to reach a final prediction is to independently run each image of a study through the same classifier, resulting in an independent probability distribution for each of them. So in this example, the classifier is pretty confident for the first, second, and fourth image, but is unsure about the third one. So we average these predictions and reach a final probability, which successfully overrides this outlier and produces a coherent final probability distribution. And to reach our diagnosis, we simply ignore the body part. So if the maximum probability was normal for arm, our diagnosis would be normal. So in this work, we will explore different scenarios and techniques aiming to maximize this classifier robustness. So our goal is to use pre-training networks as a baseline for our work, but these networks expect square inputs. So in our first experiment, we will decide on how to transform our image into squares. Our first option is to fit the image into a square, which results in cropping the center part of the image and eliminates part of the image, but does not distort it. The second option is to pad the image with black borders, which adds some useless data, but preserves the original aspect ratio and does not crop anything out. The third option is to stretch the image into a square and distorting the aspect ratio, but including the whole image without wasting part of the input. So to decide on the best option, we train the ResNet 152 network on the 3 transformations and reach these results. This gives a slight advantage to the stretch method. So this is the one we will use for the rest of this work. And now that we have decided on the best method to transform our image into squares, we can try other pre-training networks to see which ones perform best. So these pre-training networks have several benefits, such as a validated architecture that has been proven to perform well on other classification tasks like ImageNet. And since we already have the weights pre-trained, we don't have to train them from scratch, resulting in shorter training time. There is, however, a possible caveat that since these networks were trained and designed to perform ImageNet classification, the features learned during training might not be optimal for medical image usage. So we chose 10 different neural networks and fine-tuned them to our data, reaching these results. The best performing one was the DenseNet 161. And for the next experiments, we will only pick the best performing network of each family to move forward with. And we are discarding the models in gray. The next experiment we made is data augmentation. This is particularly important for medical applications, as medical data is scarce due to privacy concerns, and labeling the data is expensive as it requires trained experts. And since the models trained with insufficient data tend to overfit, this technique should reduce overfitting and improve the model capacity to generalize. So we tested two different augmentations techniques to see which one performed better. The first one is simply applying horizontal flip to the image and doubling the number of samples. And the second one is the same horizontal flip combined with 5 random crops, resulting in 12 images for each original image. And these are the results we obtained when using these two techniques on the DenseNet 161 network, which was the best performer in the previous experiment. And surprisingly, despite the augmentation B having 6 times more data, it performed worse than augmentation A. So we will move forward with augmentation A for the next experiments. So here are the results of the augmentation A applied to all the networks selected in the previous experiment. And they all had a performance increase when comparing to the non-augmented dataset. Now, while the DenseNet model was the best among the ones in the previous experiment, it doesn't mean that the other models can be right when DenseNet is wrong. So in this experiment, we will try to combine these 5 models in different ways to try and extract the best in each model. So if, for example, for a specific input, DenseNet is wrong, but the others are right, we can still get a correct prediction in the end. However, the individual models present a hard to avoid overfitting problem, leading to near 100% accuracy in the training set. So we cannot find the best way to combine these models if any combination will lead to near 100% accuracy. Therefore, we must fit the parameters of the ensemble using the validation data. The first approach we tried is very simple and based on voting. So the most predicted output becomes the final output. In this example, models 1, 2, and 5 agree with each other, while model 3 think it's an abnormal elbow, and model 4 think it's a normal humerus. So our final prediction would be abnormal humerus. In the second technique, we take into consideration the probability distribution, and we average them using different weights for each model. So in this example, we use the weights 40, 30, 20, 5, and 5. So the models 1 and 3 agree with each other, and models 2 and 4 also agree with each other, while model 5 predicts something completely different. If we didn't have the weights, we would have a final prediction very close to a tie. Since models 1 and 3 have a combined weight larger than 2 and 4, the final prediction clearly favors models 1 and 3. And since we only have 5 weights, we can find these weights using an exhaustive search instead of gradient descent. So our third and fourth techniques use a neural network on top of the other neural networks. The first one is a sparsely connected neural network where the final probability for a class is based only on the individual probabilities for that class. While in the second one, a fully connected neural network, the probability for each class in the final probability is a combination of all the probabilities in each individual model. And our last technique is to use a support vector machine to combine the results from the 5 networks. As we have 5 networks and 14 classes, so each point has 70 dimensions. So this image is a 2-dimensional simplification for illustration purposes. But the idea is the same, that we use this support vector machine to find the hyperplanes that best separate the data from different classes. And these are the results for the ensemble models. And the support vector machine was the best, but interestingly, the performance of the weighted average, the sparsely connected, and the fully connected techniques didn't correspond to their complexity. We should note that the sparsely connected is the fully connected with some weights equal to 0, and the weighted average is the sparsely connected with some weights equal to each other. So if one is a special case of the other, the performance should increase instead of decreasing. And actually, it does increase in the individual image classification, but when we group the image from the same study, the performance reverses. And here's an example where we can see how the ensembling helps improve the classifier. In the middle are the GradCan heat maps for the 5 individual models, and at the bottom is the average of them. The first 3 networks predict correctly, while the last 2 predict incorrectly. We can see that ResNet completely misses the fracture, and DenseNet focuses a little bit too much to the right. But the ensemble model, however, can take all models into consideration and outputs a correct final prediction. So to summarize, here's the final methodology we chose based on the kappa performance. The original image is stretched into a square and processed by 5 different neural networks, which were trained using an augmented dataset. And finally, we combine the 5 predictions using a support vector machine. And we repeat this process for each image of the study. So here's an overview of all the results from the experiments on the validation data. We started with the lowest kappa as 0.62 and reached the highest as 0.73 using the support vector machine ensemble. And now we transition to the test set, which is completely isolated from the validation and training sets. So on absolute terms, the best performing classes were elbow and wrist, but when comparing to human radiologists, the best ones were actually elbow and finger, where we were able to match the human performance. And expectedly, we do have a performance drop between the validation and test set. So this performance drop we just saw is likely due to overfitting of the model to the validation data. So to verify this, we can run the test data on our consensus model, which doesn't have any extra parameters. So it wasn't directly trained using the validation data, and therefore is unlikely to overfit this data. So here we can see that this model actually performed slightly better on the test set than on the validation set, and also better than the support vector machine in the test set, which in the validation set was way better than the consensus. So we can assume that if we are able to reduce overfitting in the individual models in such a way that we will not need to use the validation data to train the ensemble parameters, we could possibly reach a similar performance between validation and test on the support vector machine model. Or alternatively, we could split the data into 4 sets. So we could use one of the sets to train the ensemble parameters, and we wouldn't have to fit any parameters to the validation data. So in conclusion, the best settings we found were to stretch the image into a square, use pre-training networks, and train these networks using horizontal flip data augmentation, and ensemble these networks using a support vector machine. Our most successful results were when classifying elbow, finger, and wrist, where in the first two we were able to match human performance. Some possible improvements to this work that would be necessary to transition this work to the clinical setting include providing translation and rotation invariance, so the output doesn't change depending on how the image is inputted. And explainability, so if the model includes an explanation on how it reached its conclusion, it will be easier for a human to tell if it's coherent or it's just a false positive or false negative prediction. And finally, using larger data sets will definitely provide a big improvement on this task, as currently overfit is still present. So this ends this presentation. Thank you for your attention. If anyone has any questions, I'll be happy to answer them now.
Okay, thank you, Guilherme, for your presentation. It was really nice and really easy to follow all your methodology. So again, we are open for questions, and everyone is, just have to put your question on the chat, and then you can go and ask for the people that are presenting now. So far, we have no question in the chat, and I hope that we can have at least one in a few hours, at least before we finish the session. Anyway, but I can make one. again, I think it was, I mean, some way, I mean, quite similar to the previous presentation. So using diagnostic on using until on the image, let's say, but different problem and different image and the different problem for classification. And that Guilherme, I was, I mean, wonder that, I mean, if I got right, I mean, it's pretty much the same problem. So people, they try to combine, I mean, they try to use, I mean, different convolutional neural networks, so different architectures. And that in the end, they come up with the idea that, okay, so I don't know that they are not able to classify, I mean, everything. So it would be better, let's say, I mean, to combine all these results. But on the same time, I mean, you are putting more complexity, I mean, in terms of doing all this calculation, all this estimation of the model. And okay, you can go like, I can put more complexity, so I can combine all these models, can have a better, I mean, performance in the end. But I mean, it's really hard, I mean, to push the explainability. So I'd like you, I mean, to talk a little bit about how can you think about using explainability of the model that we're proposing right now.
Yeah, so I guess the currently model proposed does not consider explainability, as it really branches into different models. So this is really a topic for future research, as I have not considered on this matter.
Okay, but let's put another way. For instance, I've seen that the first, I mean, in the first attempt, I think that you realized that the density net, I mean, the density net 161, they provide a kappa of 0.67. So it was really, I mean, not that good, but at least was the first attempt. And then you went to the data augmentation, and the data augmentation using the A scenario that you present, I mean, increased a little bit. So it was like a 0.684, so it's less than 1/10 points, I mean, decimal point in terms of improvement. So it was not so good, let's say. And my point is that, do you think that will help, I mean, in the end, if you are not, I mean, let's say, in terms of explainability, we know that we are using combining different neural networks, and in the end, you have to open the box of each one of these models, and in the end, to be really hard. And how would it be, for instance, if instead of running and combining all these architectures, try to find a way, I mean, to improve one of these architectures, let's say. I think that you put an interesting point in here, that you said that if I using pre-training models, and that those pre-training models has been, how can I say, stressed using ImageNet, probably they are not so good, I mean, for medical diagnosis, I mean, for image diagnosis, because, I mean, in the end, the filters and the convolution, they are pretty different in terms of the objects that they have to come up with these images. So my point is that, do you think that would be, I mean, interesting to explore, I mean, going deeper in each one of these architectures? Let's say that the one that you have pretty good results, the density net 161, and try to stress more, I mean, the architecture, or either go back and, let's say, training all the stuff, I mean, build it from the scratch and not using pre-trained. Do you think that using pre-trained would be, in the end, provide good results?
Yeah, I think so as well. The amount of data we have is really limited compared like to the ImageNet data set. So if we had trained them from scratch, it would be like we have more overfitting, and in the end, I believe the results would be worse if using training from scratch instead of the pre-training, despite the possible caveat of having features that are not optimal for this task.
Okay, but let's say that we have a build, I mean, we have a data set that are based on only images for medical diagnosis, for medical image, and that pretending, I mean, using this medical image. I think that would be better, I mean, in terms of overfitting that we're talking right now. I mean, if you work with a bunch of lots of images, but in the end, they are not, I mean, the same filter or not the same, I mean, how to say, pixels that they come up with the image that can sort of have a, how can I say, images that are clear, I mean, easy for the classification method using the medical assistance, like this one that you are presented right now. I disagree, because I think that you see when we go with, and you can see that, I mean, from your pretending for the first, points that you present, in a way that you come up with the best model, just, got 0.67. So I think that there is something that you can, I don't know, and can play, either in pretending or I don't know, that can help you to stress out the architecture of these CNN. Because in the end, it's something that you will realize probably when you go for explainability. But again, it took a lot of time. I agree. If you are trying to build all the models from scratch, but in the end, if you have a good, image database that's pretty, similar to the problem that you are facing right now, chances are that will be the best model in the end. So maybe you can use, a soft architecture, not that so fancy, like, let's say, like this one, because you have a good database, you have a good data set. So this is always something to bring the discussion. But it was really nice work, and I think that you did all the stuff. The only thing that I usually question people, it's about the baseline, and there's something that you bring in the discussion, that you said that for some of your results you compared with the human, and the result was pretty good. Would you like to talk more about this? How can you evaluate this, comparing your models against the human doing the same classification?
Well, first about the pre-trained networks. So we did try another approach, which consists of pre-training the models using another medical data set a bit larger, and then fine-tuning to our specific problem. But the results were worse than using the ImageNet pre-training. So we abandoned this approach. And what was the other question?
Oh, it's okay. It was based on discussion. But I was, a little bit skeptical about the using the retained image, from the ImageNet for all stuff. I think that there are lots of people using this for many different computational visual problems. But in the end, you see that all the time what people do is, in the end, they combine many architectures that are available. So let's say that they have, I don't know, 10 huge, how can I say, TNNs, and then they combine in the end. So what I'm saying that, what if in the end, the problem is that the data set, because that the model in the end, they rely on the data set that you have. That's not a miracle. That the same thing that people used to say, that garbage in, garbage out. So if you have poor database, the data sets in the end, the model to be pretty much, bad. So the problem is, how can you use it? Is in the smart way, the data sets that are available. I think that there are lots of interesting discussion, and I'm just putting this in discussion to you, so in the way that to push you, to go deeper in the CNN architectures, and going back with I don't know, data augmentation. You present the data augmentation help, but not help that much. I think that you're probably expecting more. And but anyway, there is no second question. It was basically one regarding this pre-trained model, the using of pre-trained model. But again, thank you very much, Guilherme, for your presentation, and it's really interesting work, and I think that there are many interesting possibilities, and we can, but the one thing that you can say that when by the time that you change the domain, for instance, for the work that Gabriel presented on COVID detection from the lungs, it's different. So maybe we can do all the stuff again, but again, at least the methodology to be pretty much the same. Okay, thank you very much. I don't know if we have, we may have, I don't know, time for one more question, but I don't know. Let's see in the chat if we have some. No, so okay. So thank you very much, Guilherme. We can go for the next presentation.
Thank you.
And I think that the next one is Jorge Francisco, that will present the combining mutation and gene network data in a machine learning approach for false positive cancer-driven gene discovery. Please, George, start your presentation.
Okay, Professor Horner. And let me share my screen. Okay, can you see my screen? Okay.
Yep, it's okay.
Okay, good afternoon. Hi, everyone. My name is Jorge Francisco Cutigi. I will present our work with title Combining Mutation and Gene Network Data in a Machine Learning Approach for False Positive Cancer Driver Gene Discovery. This is a collaboration work with researchers from Federal Institute of São Paulo, São Carlos Campus, University of São Paulo, São Carlos, and Barretos Cancer Hospital. First, I would like to thank the organizers, the BSB 2020 organizers, for the opportunity to share our work in this important event. Thank you for the audience for being here virtually, watching our presentation. Thank you very much. So this is the agenda of our presentation. First, we'll talk about the context, the problem statement, our objectives, followed by our proposed method, our computational approach, machine learning approach. Next, we'll talk about the results with a model evaluation and a concept application validation. And finally, a conclusion of our work and results. So, about the introduction, cancer is our object of study and is a term that describes a large number of complex diseases. There are many types and subtypes of cancer that happen in many parts of the human body. Cancer is known to be caused by alterations on DNA. That is, cancer is a genetic disease. These alterations, they are known as mutations. And in the context of cancer, mutations can be classified as significant or not significant for cancer. The significant mutations for cancer, they are involved in the initiation and progression of cancer. The significant mutations for cancer, they are widely known as driver mutations. Not all mutations in a cancer cell, they are drivers. Just a small number of mutations are drivers. No? And when such mutations, they are related with genes, these genes can be called as driver genes. And so the identification, if the driver genes, they are identified, if we know which genes are drivers, it can be used to assist on cancer diagnosis, prognosis, and personalized treatment for the patient. And the identification of driver genes, it may be supported by computational methods. And many computational methods have been developed in past years. These methods, they are based on various types of data that are currently available. For example, these methods, they are usually based on mutation data that are NGS sequencing data, where we have the tumor, a cancer cell, and the genes and the mutations that tumor, the genes, the tumor and the genes were mutated genes in that tumor. Okay, and another example of data is the gene interaction networks that are complex gene interactions among genes and their related proteins. And they are modeled here as complex network. So here is the problem. Some genes, they present characteristics of drivers, but they are not drivers. These genes, they are not involved in cancer initiation and progression. And the computational methods, they can misclassify some genes as drivers. And these false positives, they are known as false drivers. The false drivers, they have an important impact in cancer analysis. They can lead to identify unrealistic trends in data.
and they can compromise the performance of model that take information of driver genes as variables, and they can mislead biomedical decision. So avoiding the misclassification of false drivers as drivers is an open challenge in cancer genomics. So in this context, we have our proposal. The objective is to develop a screening tool to discover false drivers among driver gene candidates. Then if I have given a driver gene candidate, we intend to classify this candidate as a real drive or a false drive. So the approach we performed to do that is to extract useful information as features for models and induced machine learning models to classify the gene. Okay, and these models we intend to induce then with a data set created from the combination of mutation data and gene interaction networks. So about our proposed method, our approach, here we have an overview of our approach in 3 steps. The first step is data collection, where we collect cancer mutation data, gene interaction networks, and the labels of genes. The second step is the data set creation. We perform the pre-processing routine in the collected data to create the data set to train the model. In the third step of the model training and evaluation, where we select 2 machine learning algorithms and we evaluate the results. So about the first step of our approach, the data collection, we collect data from 3 sources. The first is mutation data, gene interaction network, and gene labeling. So we have 3 sources to collect the data. The first, mutation data, we select cancer data of 33 types of cancer. They were selected based on a recently TCGA pan-cancer study available on TCGA website. 2 types of mutations were considered. In our case, single nucleotide variants, SNVs, and insertions and deletions in those. The second source of data are the gene interaction networks. We select 4 networks, 4 widely used and known networks. These networks, they were combined into a single gene network. So we have here a consensus network. Okay. The last source of information is about the labels of the genes. We have here labels on genes for possible false drivers and drivers. We collect this information from NCG, and CGC to reliable source of information about genes and cancer. So the second step of our approach is the data set creation. First, we performed the cancer data pre-processing, and we first removed the hypermutated samples. We considered the hypermutated samples as outliers, and we removed the samples in the patients where the number of mutations they have are much higher than the average. So we removed this outlier, these hypermutated samples, okay? And we also kept 9 specific somatic variants that are related with the coding regions on DNA. For example, frame shift del, missense mutation, non-stop mutations, among others. So after the pre-processing routine, we have this feature extraction. From cancer mutation data, we extracted 10 features. One feature is the number of patients that the gene is mutated and the number of mutation of each somatic variant. So we have here 10 features. It generated for us a data set, a mutation data set with information about the mutation of the 33 types of cancer. From the gene interaction network, we also extract 10 features. These features, they are related to centrality measures, node centrality measures from a network. For example, degree centrality, betweenness, closeness. It generated for us a data set, a gene network data set with genes and information about the gene centralities on the network. Here, we have the combination of the mutation data set and the gene network data set, and we generated a combined data set with information about mutation and gene network. So we have here a labeled data set of mutation network data with 20 features, 10 from mutations, 10 from gene networks, and 876 samples. Into these samples, 647 are drivers and 229 are false drivers. These are the labels of the genes. So with this combined data set, we can go to our third step of our approach. It's about the model training and evaluation. This was a machine learning training process. About our predictive models, we selected two machine learning algorithms to induce the models, the predictive models. They were support vector machine, SVMs, and random forest. The models, they were induced using a stratified 5-fold cross validation. It's in order to avoid the overfitting, and undersampling to address the class imbalance, because we have a driver as a majority class in our data sets. We also performed a hyperparameter selection. We evaluated it, we assessed it by training the multiple models in a grid search process. We repeated each models 10 times to account for the possible influence of randomness, both for the random of the models and for the class imbalance, the undersampling. Continuing here, the evaluation, about the evaluation and validation criteria, our objective is to evaluate the predictive potential of the model. And we intend to evaluate the performance of a model induced using combined features in comparison to a model induced by a single source of features. For example, we want to compare the models, the results for the model using combining data versus mutation data, and combining data versus gene network data. And the analysis were performed using some classical measures, accuracy, precision, recall, F1 score, and ROC curves. So now let's talk about our results. First, about the model evaluation. Here we have the results for SVM models. Then we have here in the rows the measures, accuracy, precision, recall, and F1 score. And we have 3 columns. The first column are the results for the models induced using combined data set, the data set with mutation and gene network data. The second column, we have the results for the model induced using just mutation data. And the third column, we have the results for the models induced using gene network data. We can see here that the results for the model induced using combined data sets have better results in comparison with mutation and gene network data for SVM. Here we have the results for random forest, the same experiment for random forest. We have similar results here. For combined data set, we have better results than in comparison with a single source of data, mutation or gene network data. So with this result, summarization, we can see here that the combination of mutation data and gene network, they improve the potential, the predictive potential of our model, both SVM and random forest. When we compare SVM and random forest, we can see here that random forest is better, considering precision, is better than SVM. For the others measures, accuracy, recall, and F1, SVM is better than random forest. Now, a rock curve analysis for SVM is a nice way to see the results here. We have the red curve is a rock curve for the modules induced using gene network data set. And the blue one is for mutation data set, and the green one for the combined data set. We can see here a better curve for combined data set. And the results, they are here. They are also here in area under the rock curve, the combined data set. We have a better area under the rock curve than mutation data set and gene network data set. This is for SVM. For random forest, we have similar results. The red curve for gene network data set, mutation data set is blue, and green, the combined data set. We have here the area under the rock curve for random forest. Comparing random forest and SVM, we can see here the transition, SVM, the rock curve, sorry, SVM and random forest, okay? And the area under the rock curve is better for random forest than SVM. So, still on results, we performed a concept application and validation of our models. And for this concept application validation, a data set composed of new driver gene candidates was required. For this, we used a subset of 131 genes that were recently reported as drivers by Intogen. The Intogen was recently published on Nature during the development of our work. It was published in July, final of July of this year. And our models, SVM and random forest models, they were applied to this subset to detect possible false drivers into this subset. Okay? And the validation, it was performed based on literature. The results of the classification of SVM and random forest models, they were compared to statistics of current literature reports using CancerMine. CancerMine is a novel tool, is a text mining tool that extracts from literature evidence of cancer genes. CancerMine classifies a gene as a driver, a oncogene, or tumor suppressor genes. That is, if a gene is related to cancer. So here we have the results for our validation. Here we have the genes classified by our models as false drivers and drivers. And here we have the frequency that these genes appeared on CancerMine for one or more citations. Here we have the same for 3 or more citations, and here we have for 10 or more citations. We can see here, we can notice a difference between the frequency in citation for drivers and false drivers. In CancerMine, drivers appear more than false drivers for the classified genes by our models. Then it can be an evidence that our model, they can likely identify false false drivers and drivers in drivers gene candidates. Sorry, this is the result for SVM. And here we have the same experiment, the same validation for random forest, a small difference between the classification of SVM and random forest. Okay. So now about the conclusion of our work, we proposed a machine learning approach to detect potential false drivers. So among driver candidates, our model can classify these candidates as false drivers or real drivers. The models, they were induced using a data set with the combination of mutation data and gene interaction networks. And this combination has improved the classification of the classification performance of our models. And these models, our models, they can be used to avoid the misclassification of driver candidates. With this, it's possible to eliminate unnecessary further analysis, and to detect false drivers can prevent their inclusion in data analysis or on the development of models that use these driver genes as variables, as features, for example. And the limitations of our approach. They classify driver genes in general, not for a specific type of cancer. So we can classify the genes for a cancer in general, not for a specific type or subtype of cancer. So thank you very much for the attention. Thank you for the organizers again for the opportunity to be here, present our work. Thank you for the audience, and I would like to thank my co-workers who worked with me in this paper, and Renato, Rodrigo, Cynthia, Adriane, André, and Adenilso. Thank you very much. I'm open for the questions.
Okay, thank you, Jorge, for your presentation. It's really interesting. I mean, we can see a really nice application on cancer study and cancer research. And I don't know, let me check again on the chat if you have any questions. No, we don't have any questions, but I'm really, excited to make some questions to you. It's really nice work. And well, first I'd like to check with you, about the data set. Again, I think that before starting doing modeling or doing machine learning, building models, I think that the first step is get into the domain and get a really nice view of what we have in terms of data set. I think that's one of the things that you did. That was let's see what we have and let's combine these and see if we succeed in the end in building a model. And if I'm not right, if I'm not wrong, you said that the mutation data set, the mutation database that is used, it's from a publication. It's from I did not guess because I'm familiar with the Cosmic database, and I was wondering about if you think that would be help in your method if we use the Cosmic mutation database, or what was your choice about not using this Cosmic database for mutation?
Okay, we collect the mutation data from a recent TCGA pan-cancer study that was published in end of 2018 in Cell, if I'm not wrong. And so we collected this data from TCGA. We didn't use Cosmic data mutation data in this work, but we could use it with no problem, of course.
Yeah, so I was just wondering because you can in some sense enlarge your data set. So I don't know. I have no idea because I never. I'm familiar with Cosmic, but I never tried to build model or whatever. But I think that's a really nice database that is available and all related to cancer research. And since they have information from mutation, maybe I don't know, it can help you on your research.
No, okay.
And I have, yeah, but
no, about the cosmic, the labels, the CGC is from the cosmic, and there
okay.
are a lot of important information there. And then from cosmic, you use for the gene labels.
Yeah,
Sorry,
I see.
interrupt you.
I see. But anyway, I mean, you are using TCGA. TCGA is the large one, the most important, I mean, huge project in non-cancer research. And I was, I mean, maybe a few years ago, I was working with this in France, I mean, for the project, and I'm quite familiar with this. And I remember that that time the point was that we were not able, I mean, to download the raw data. And for this project, we were really interested in having the raw data to all pipeline, I mean, to treat the data and reprocessing all this stuff. Because I mean, if you're not, I mean, subscribed as one collaboration with CGA, you don't have the, you only have the final data set, let's say. So all the things that you can do, I mean, for pre-processing, you are not able to have this. And I have another question that is related to one really interesting point. That's how can you deal with unbalancing data sets? And you said that you have been using undersampling, so I think that was handle undersampling. And have you tried to apply SMOT, for instance? That's another approach for handling imbalancing data sets. Call it SMOT. Have you tried?
Yeah. Okay, that's a nice question. We use the undersampling. We just randomly removed the majority class in each fold of our process. We tried to use another approach to deal with class imbalancing, for example, oversampling. But with undersampling, we have the better results, and we believe that we decrease the possibility of bias on data, because, for example, the oversampling, we tried to duplicate some samples. The result was not so. The results was good, but the undersampling was the results were better.
Okay. Yes, it's really. I have another question. Again, we have a time, so I'm trying to have this chance. I mean, for have a chat with you, because I mean the other guys. I mean, they are kind of I don't know, timid to make more questions. But I mean, all people are invited to make questions on the chat, and then you can follow for the people that are presenting right now.
Okay.
Again, the last question is I've seen that you use endophoric SVM, and have you tried to try all the models like for instance gradient boosting machines, GBM? It's really, I mean, people have been using a lot of this kind of application, and I was, I mean, interested. I mean, if I have plans to try to check, I mean, GBM, because I mean, in the end, when we look to the all AUC, I mean, results, it's pretty much the same, you know, for SVM and endophoric. I think that if you put this on a test, let's say if you put this like a statistical test like Friedman and any other post-hoc analysis, and you see that they are not different at all. The difference that's not statistical, you cannot have a confidence about the difference because it's really small. But again, it's something that you have to check and have to try to make sure that they are different or not, and the result. But again, do you have any plans to building or trying other methods, let's say other samples or combination?
Yes. We have plans to extend this work and try other things to try new models. In this case, we didn't try other models because we have chosen SVM and random forest because we didn't know how the problem could be modeled. So SVM and random forest, they are flexible models. It's possible to from the hyperparameter adjustment, we can vary the complexity of the model architecture. Then that's the reason we use SVM and random forest. But we have plans to extend this work and try another interesting models and another process and another features. We have a lot of ideas to this work. Thank you very much for the suggestions.
Okay, George, that's really nice because as I said for the other guys that presented before you, I mean, usually, I mean, sometimes it would be better when we have this idea of baseline. So let's say you present a problem and you say, okay, in this problem, the guys usually they have like, I don't know, accuracy of 70%. So that's the baseline. So everything that they can build up a little better than this, it's nice. I mean, it's great. When you present like a handle forward SVM, we have this, I mean, I don't know, wrong idea that the challenge is really high, but no, because there's something new that you're providing, so trying to use a simple regression, for instance, and let's say that, okay, my baseline is the simple regression that I have these results. And then we go, I mean, using more sophisticated methods because it is, as you said, I mean, when you put complexity on the point, I mean, you have to balance. I mean, if I use this and that, I mean, on one hand, I mean, I think that's something that we've been discussing during this previous presentation, that from one side, you have the performance in a way that, okay, they go combining different stuff, different methods, and again, you have a really nice performance, but you have no idea, I mean, how the algorithm took decisions. So from the side of explainability, it's a huge gap. And we know that nowadays that people are stressing a lot about this. So we have, I mean, now, I mean, we like people that work in this machine learning, that we have to be aware about this. So in balancing this complexity and interpretability. And that's something that you have to take into account. Okay. I think that Valdeir Mendes have a question. Valdeir, we have a time, please.
Hello, Jorge. If I understood correctly by the previous question, you normalize the data before, right? Because you have a lot of different kind of cancer drive genes to give as input, right? And my question is, after all of this, when you look back to your results, could you identify if there is a particular type of cancer that is where the results are better or worse?
And okay, Valder, thanks for the question. We didn't work with a specific type of cancer in this case, just in a general case. Okay, then this is another ideas we have here to deal with a specific cancer type or subtypes of cancer. Then in this case, we didn't have a result for a specific cancer type. Then I didn't have this answer. I don't know if I answer your question.
Yeah, but this drive mutations and genes are you cannot identify how are they related to a particular kind of cancer. So you cannot extract this information from your results.
No,
no. Okay.
No, the drivers they can be. There are drivers that are common. They are common for the cancer. For example, TP53 is a common driver for all types of cancer. But there are specific drivers for a type of cancer. But our work, we didn't have this information, these results from our models. Okay.
I see. Thank you.
Thanks for the question.
Okay, thank you, George. Thank you, Valdeir. We have another question from Gerda Cristal. So she asked about, do you perform gene ontology analysis or with the selected driver gene? So it's pretty much in the way that you showed about the Taximine tool and trying to balance the results. Can you talk a little bit more about it?
Yes, that's a good catch. No, we didn't perform a GO analysis. The concept application validation, we performed using cancer mine as the validation as presented. But that's a really good idea and important to see the biological results of our work. But we didn't perform this geo analysis. Thank you, Crystal, for the question.
Okay, so thank you again, George. It was really nice discussion and presentation. And then we can go now for the next presentation.
Thank you.
That would be, let's see, Matheus Ferraz. So the presentation should be about revealing the role of nanobaric threads on their folding and stability assisted by machine and deep learning algorithms. Okay, Matheus, we are ready for start.
Can you see my screen?
Yes.
So let's go. My name is Matheus, and I'm going to present my work entitled Unraveling the Role of Nanobodies Tetra during the Folding and Stability Assisted by Matching and Deep Learning Algorithms. I'm a PhD student in chemistry at the Federal University of Pernambuco, and I perform my research at the Oswaldo Cruz Foundation in Pernambuco also. So the main idea of this work, it was to assess biophysical features of nanobodies assisted by machine learning methods. But what actually is a nanobodies? Well, I guess I'm going to shift a little bit the presentation topics to chemistry sometimes, but let's go. The nanobodies are proteins that, in addition to conventional antibodies, some commonly the species they can produce, those nanobodies molecules, they are naturally produced, they are naturally induced when a camelid is in contact with an antigen. So those are antibodies devoid of heavy chain. They have only, those are antibodies devoid of light chain. They have only heavy chains, and they don't have light chains. And this fragment here, which we call VHH, which is the variable heavy fragment, we call it nanobodies. And the nanobodies for themselves, they can fold properly, and they can bind antigens, and they are highly stable and highly soluble. Besides, they have low production cost. So when you think of producing and buying antibodies, it's really, really costly. But when you think of producing nanobodies, that's not that costly, and you can produce it in large scale in bioreactors. So together, all those features makes the nanobodies strategies for therapeutic and diagnostic innovation, since they have low immunogenicity. And for now, there are many, many nanobodies under investigation against HIV, against cancer, against coronavirus, against Zika virus, dengue, and many of them. And last year, the first nanobody was approved. So just looking some general features of the nanobodies, here we compare a nanobody and an antibody. As I told, the nanobodies, they won't have the light chain, only the heavy chain. And since they don't have their partner here, they are now exposed to the solvent, exposed to water. Thus, nature has driven the design in such a way that has proposed 4 polar residues in the nanobody surface. They're located here on the surface, and they're highly conserved in nanobodies. And they are there because the nanobodies, they don't have the light chain, so they need to have some changes in the structure, in the sequence, to be exposed to the solvent. And we know that the nanobody, the 4 residues, they're called the nanobodies tetrad. And we know that these residues are responsible for giving the nanobodies their stability and solubility. But we don't know the reason. I mean, what's the chemical force that drives the nanobody stability and solubility? We know that it's the tetrad, but we don't know what's the reason for the tetrad being there. And this is really important when you think of engineering proteins, when you think of designing novel nanobodies. Since nanobodies, they have a wide range of applications in the biotechnological industry. They can be used for many applications, therapeutics and diagnostic. And one of my main research topics during my PhD is to design specific nanobodies computationally. And the first nanobody we have designed, we have moved completely the tetrad. We have completely masked the tetrad, and it changed it completely. And the result was that our nanobody aggregated. So it was not stable. So we saw there that nanobody's tetrad is really important to be there. And since we changed the tetrad, it was completely aggregated. It didn't have a three-dimensional fold. And then this nanobody, it was like not good enough. Since it aggregated, it was not stable. And we changed the tetrad. So we thought of understanding the role of the tetrad by using molecular dynamic simulation. We had a small set of 3 nanobodies. We had the experimental stability determined, the binding energy of stability was determined. And when you use molecular dynamics and energy functions, we could have the same trend using both techniques. But when we use it, I know the color was not a good choice, but this is another protein that's not this black one here. But when we try to use the molecular dynamic simulation to study a novel nanobody that aggregated, this one here, we saw by molecular dynamic simulation that it was really stable, and we didn't expect that. And when we went to literature, we saw that actually molecular dynamic simulation can't help understanding the stability of nanobodies, and you can't predict whether a nanobody will be soluble and stable or not. So we thought of novel approaches for designing nanobodies. And for this approach, we have designed nanobodies considering the tetrad. And as for a result, we have been designing nanobodies against coronavirus, not altering the tetrad. And here are some preliminary results where we can see that nanobody was completely stable, it was completely soluble, and we have expressed it, purified, and we're right now conducting the neutralization tests to verify whether this nanobody can be used as a neutralizing agent for coronavirus. And we have also performed some enhanced sampling molecular dynamic simulation to understand, to predict whether this nanobody would bind to coronavirus or not. And I hope so that it will bind. Theoretical results are really promising, but we have to wait for experiments that are being conducted exactly right now. But yes, we show that the tetrad is important. When we remove the tetrad, we don't have a good nanobody, but when we leave the tetrad there, we have a good nanobody, but we still don't know what's the reason for the nanobody's tetrad assured solubility and stability. So to this end, we thought of using machine learning to try to understand. But before applying machine learning, we need to ask ourselves, how can we featureize a protein data set? I mean, there are many protein data sets available, and there are many models of machine learning using these data sets. And these data sets, they are mostly based on sequence, but you're not interested in the sequence, you are interested in the structure. So how can we describe protein structurally speaking? So far, I have not seen no one using this approach to describe the proteins. But when you think of describing a molecule, we usually think of using a quantum equation. We usually think of a Schrödinger equation to describe, but it's not feasible for huge systems like proteins. So we're using classical atomic potentials that describe the interactions within a protein, be it their bound distance, their angle, they perform, the dihedral, the electrostatics. So we can use those classical potentials that describe the physics of a system to featureize proteins. And to this end, we are asking ourselves, what's the role of the VHA, the Nanobots tetrad? So we had two data sets. It was really a small data set because that was available. But then we know we have the Nanobots tetrad, and we replaced the Nanobots tetrad for alanine residues. But why alanine? Because it's the smallest amino acid, and it has a massive group here. So it does not make any significant interaction. So we divided, we went to the protein data bank database. They have downloaded 30 structures of nanobodies. I know that's very little, but that's what's available. And from the 30 structures, we replaced it for all of them, the tetrad for alanine. And had now 30 native nanobodies and 30 engineered nanobodies using the software Rosetta. Of course, after doing this replacement, we minimized the geometry of the structures and we calculated potential energy terms using the Rosetta energy function, which I'm going to mention in the next slides. And we have used 3 machine learning models, SVM, random forest, and multiple layer perceptron to try to identify if there is any feature between those two classes that would explain the presence of the nanobodies. When you remove the tetrad, what does it happen to the structure of nanobodies? And when I think of the Rosetta energy function, I'm thinking of physical potentials to model molecular interactions. So we use actually chemical physical theories like electrostatics, Lazarus decoupled equations for salvation energy, energons to the van der Waals. And we also have energetic terms conditioned to protein structures, like the backbone torsion energy and hydrogen bond. Those properties, they are extracted directly from the protein data bank. So we calculate all all the potentials using the energy function of Rosetta. These are all the features we have used for within the Rosetta energy function. And we tried to use these models to identify features that could discriminate between the native nanobodies and engineered nanobodies, so that we could think of the role of the tetrad within their nanobodies. And first of all, we have used extremely randomized trees methodology to perform a feature selection, since the Rosetta energy functions contain all these 18 terms, and these terms are related to the energy observation, they're related to the proline angle, to the Ramachandran angles, to hydrogen bonds, to attraction and repulsion, and all these are physical terms related to the protein geometry. We actually do not use the three-dimensional structure of the proteins, but we rather use, we project the three-dimensional features in these attributes. And our first guess was to use a linear discriminant analysis so that we could verify whether there is any significant difference between the nanobodies. And what we see here is that the linear discriminant analysis actually can discriminate satisfactorily the natural nanobodies and the engineered ones. So here in blue, we have the natural, and in red, the engineered. And we can see that linear discriminants can discriminate those two classes. And when we go to the linear discriminant loadings to see the influence of each variable we have considered, I have considered only features possessing a feature importance greater than 0.005. So I have those features here. And the most significant ones were the salvation energy, which we call here the LK ball, and the reference energy. The reference energy is a value that tells you how that will model the unfolding propensity. So if you have a structure with a high ref, it means that your protein has a high propensity to unfold, so it's not stable. And another one, it was the F8 done. The F8 done is a metric that tells you about the distribution of the dihedrals of angles in the protein. And when we compare these features, the average of those features for the 30 nanobodies, for the 60 nanobodies, and we split them into the native, the natural ones, and engineer it, we see that they have highly significant difference using a t-test, an unpaired t-test, and we calculated the p-value, and p-values lower than 0.05 were considered to be significant, statistically significant. And we see that actually that our groups, now we have a binary classification of natural and engineering, they actually do have differences in some features. And those features, they might explain the reason for the stability and solubility of the nanobodies. And then we have used 3 different machine learning models to identify if we could, to assess the performance in separating the both fats of nanobodies. And what we realize here is that as SVM and MLP, they have good performance, but random forest performance is really, really poor. I mean, the standard deviation is like higher than the average value. And we can confirm the validation score, the accuracy of their models, using the confusion matrix, where we can see that SVM can predict quite accurately, and MLP has also some good performance, whereas for the random forest model is not that good. So we think that the classification task is not really straightforward, as it's not straightforward. So it might require some model that's more robust than the random forest. It's much simple. And then we have also assessed the rock curve, and the rock curve, they just reflected the previous behavior. And most importantly, we also looked to the learning and training curves to verify whether it was happening over 50 or not. I know that the data set is really small, but then I wanted to see how the score compares as we increase the number of examples, of number of training examples. And what we can see here is that for SVM, we have a training score that's decreasing, and the cross-validation, the test score is increasing. And I mean, if we increase the data set number, I expect to see increase also in our cross-validation score. As for random forest, the cross-validation score is really decreasing all the time, and it has unsatisfactory values. And for the MLP deep learning model, we realized that it's increasing also, and that our training score was kept constant. It indicates that we are having, yes, an overfit of the data, but that might be due to the small number of training examples, that it was a limitation of the problem, because we use the available structures from the PDB data bank, and it couldn't increase it more than that. And as conclusion, we see that the 2 classes of nanobodies, they differ mainly by folding and salvation metrics, as we have seen using the LDA approach. And we just highlight the importance of the tetrad for stability and solubility, and that this stability is achieved mainly due to entropic contribution. As I've mentioned before, the 2 metrics that most impact in separating the classes were salvation energy and the distribution of angles. And all those 2 features, they are completely related to the entropic contribution of the energy. So I would like to acknowledge my research group and all the funding agencies that's been sponsoring the work, and the persons who collaborated within the work. And I'd like to thank you for the invitation and thank you for the organizing committee. Thank you.
Okay, thank you, Matheus, for your presentation. It was really interesting to see this featureization and how can you find at least a feature that can help you to transform your problem like a machine learning problem. Okay, let me see again if you can if you have any questions in the chat. No, I have no questions so far in the chat, but I can make some. If I understand correctly, I mean, I think the first question that related to the way that you did this, how can I say, feature, find the features. You know, let's say, how can you find the features for your problem? And I see that if I'm not wrong, I mean, you have like 18 features. So you come up with 18 features, and then you choose a cutoff that in the end you left like 7, 6 features. So the point, I mean, what was the criteria for choosing this cutoff for the best feature, let's say? Among these 18 features, you find that 7 or 6 it was better than using these 18 features. Have you tried to use all of them?
Yes, first of all, I tried using all of them, but the results, they are not that good. I mean, I couldn't separate between the classes, and the LDA didn't show a discrimination. But I then used the extremely randomized trees algorithm to perform a feature selection. Since the extra trees classifier is not deterministic, it is stochastic, I have run the algorithm for like 10 times and used the average for each run of the algorithm and selected the ones that presented feature importance greater than 0.05.
Okay, so if you use a feature selection approach before going to all the models like you presented, like FDM, MLT, and so use the extreme decision tree. And have you tried, for instance, have you used some kind of a grid search to check the hyperparameters? I mean, or
sorry, have you tried what?
Have you tried some grid search approach, I mean, to test all the hyperparameters? Because each one of these algorithms, they have hyperparameters. So the parameters that have to trickle, I mean, to tackle in the algorithm in order to have boundary, I mean, to have a good decision boundary yes. From the algorithm. So the point is that have you tried to, or have you used some kind of a grid? Because I mean, it was really interesting to see on your results that Nanobots didn't get really good results. I mean, and then there are many possibilities. One, I think that's because the data set is quite small. With data set, I mean, with that size, I mean, it would be really hard to have a good classifier. And I mean, in the sense that you can use these in the real application. But I think that, I mean, if you play with a good grid search algorithm with checking and turning all the hyperparameters, I don't know, maybe we can have, I mean, some more fair results. I don't know. I mean, my point is that if you have tried some grid search, I mean, checking the hyperparameters, each one of the methods that you present, like SD and Power. Have you tried to use some kind of a grid search?
Yes, next question. Yes, I have used the grid search based method to identify the hyperparameters for the algorithms I have used it. But yeah, so that's like a great suggestion.
Okay, so I think that we have another question here from the audience. Valdeir, he asked about are the natural nanobodies. I mean, I think that you can ask him here, Valdeir. I know that you're online. Please. I think that he doesn't.
Okay, I'm here. I'm here.
Okay.
No, it's most a curiosity about your raw data. Are this these natural nanobodies that he used to make these mutations and create the other ones, are they derived from camelids as the original ones?
Yes, actually, all the nanobodies, they are produced by camelids. Of course, we can have some synthetic ones, like the ones I computationally designed, but normally the ones in the PDB, they are extracted from camelids. Some of them from alpaca, from llama, from dromedaries.
Yeah, I see. And because of that, it's another curiosity, is the toxicity between the features, because I do not recognize the acronyms in your features. All of them are biophysical or biochemical features, or do you have this list? Some of them I do not recognize, so I was wondering if you have some feature related to the toxicity of the nanobodies to evaluate it in the mutated ones.
No, actually, the information we do have about toxicity.
No, okay. Yeah.
Usually they're not immunogenicity because they share a high homology with the human heavy chain antibody. So it's quite similar to human antibodies, they have chain. And all those features here, they are based on biophysical characteristics of the three-dimensional structure. For example, hydrogen bonds, the omega is the omega angle in the peptidic bond. The attraction term of energetic attraction, energetic repulsion, and hydrogen bonds, and disulfide bonds energy, electrostatics, solvation energy, they're all biophysical terms.
Thank you.
Okay, so thank you again, Matheus and Valdeir, for the questions, and we can go for the next presentation. I think it's the last one. Thank you, Matheus. So the last presentation is, I think it's Danielle that you presented. It's for the paper entitled Experience Data Analyzer for Runtime Analysis of Phylogenomic Data Flows. So you can go, Danielle.
Okay, can you see my screen? All right.
Yep.
Okay. Thank you. So thank you, Ronnie, for the introduction. I'm going to present the paper entitled Experience DF Analyzer for Runtime Analysis of Phylogenomic Data Flows. This is a joint work with Luiz Gustavo Dias. He's a PhD student at Fluminense Federal University, Professor Marta Matoso from Copo FRJ, and Professor Bruno Lopes from Federal Fluminense University as well. So the context of our work is focused mainly on phylogenomic workflows. So phylogenomic experiments provide the basis for the evolutionary biology inference, you know that. And many of these analyses, many of these experiments are data and CPU intensive. So they require a lot of CPU processing, a lot of CPU capacity to process results in a feasible time. And commonly, these experiments, these workflows, they have to be executed in high-performance computing environments, such as a small cluster or a grid or a cloud, to produce results in a timely manner. So one way to represent these experiments is to model them as a scientific workflow. What's a scientific workflow? It's a graph where the nodes are the activities, what we call activities or activations. So they are associated to programs or scripts or web services that we can execute for providing some parameter values to configure that activities. And the arcs of the graph are the data dependent. So in this example here, a simple example here, I extracted that from the Taverna website. Taverna is a workflow management system. We have some activities in green and pink here, and the arrows here are the data dependent. So this green activities, the invocation of our web service here, depends on the data produced by the pink activity executed before. And these workflows, these experiments, they can be represented and implemented in many ways. Common ways are representing them, modeling them in workflow management systems that are systems that provide the framework to model the workflow, to execute the workflow, to monitor the workflow, to capture results. Recently, we started using data-intensive and scalable computing frameworks such as Apache Spark or Hadoop to model and to represent phylogenomic experiments. But most of the users that we have contacted and that we have worked with, they still use their own scripts to model the experiment. Because, for example, in the workflow systems, we have some limitations about how can we represent the experiment. For example, if we have a loop, some of the workflow systems does not support loops. And using data-intensive scalable computing frameworks such as Hadoop, maybe it's hard to model the workflow, or maybe it's hard to invoke third-party code or black box applications. So most of the users, most of the bioinformaticians still use their own scripts, or part of the experiments are more or less their own scripts to execute. And these experiments require runtime data analysis, so that the scientists has to access the data that's produced during the experiment, the execution of the experiment, and they have to that an online way at runtime. So if the scientist checks if a specific result is not what he or she expected, so he can stop the execution, he can change some parameters. So it's important to do that analysis at runtime, because these experiments are CPU intensive and can execute for many hours and even many days, depending on the amount of input data. So our idea in this work is how to support the scientists to do this runtime analysis, and how we can help the scientists to achieve this runtime data analysis of the phylogenomic experiment. We use the phylogenomic experiment as a case study of our proposal. So to achieve that, we have to collect one type of metadata that's called provenance. So the idea is to use provenance as a solution to foster reproducibility. So we capture what activities were executed, what are the used parameters, and what are the errors, the produced data. So the idea is to collect provenance data to describe what's happening to the data flow, to the workflow, so we can help the scientists to analyze and to achieve some results and to confirm or refute his hypothesis. So to explain what's provenance, let us take an example from the arts domain. I don't know if you remember this case. It's a restoration performed by Spanish on this painting. So it results in this second painting. If we don't know what's happening to this during this process, it's almost impossible to associate this second painting with the first one. So the provenance is all the registers, all informations that shows that this first painting, this first picture, after data transformation, a catastrophic data transformation, produced the second one. So we can use that on the phylogenomic, on the bioinformatics domain as well. So if we are producing, for example, phylogenetic trees, it's important to map and to trace back all the information that produces that phylogenomic tree or phylogenetic tree. So it's important to know what were the input files, what were the input sequence that generated that tree. So the idea here is to collect all this provenance data, all this historical information, to allow for the scientist to perform his or her analysis. But collecting provenance data, and we call it domain-specific data, so we can extract information within data files, within produced data files, is still challenging. Mainly because adding a provenance capture mechanism can add a high overhead in the experiment. So if the experiment already takes hours or even days to execute, we cannot add more time for capturing provenance, because the scientist doesn't want that. It already implemented his code or her code to be performatic. So if we add more time to collect provenance, normally the scientist doesn't want that mechanism, because it's almost making the experiment worse, perform worse. So one challenge here is how to capture this data, how to capture provenance data without adding overhead, performance overhead over the experiment. And another problem is when we perform automatic provenance data capture, when we capture all provenance data, maybe the volume of captured data is so high that the scientist doesn't know how to analyze that, or one maybe problems to analyze that because of the high volume of data. So our idea is to provide some flexibility to the scientists, so the bioinformatician will show what I need this information, that information, and the others I don't want. It's not important to me. It's like when we go to the market. So we pick up on the shelves the products that we want. The idea here is to provide a way for the bioinformatician to define what type of information he or she wants to capture, and then he or she will use on the analysis. So there are some approaches that already provide that. Most of them are specific for programming languages, so for some programming languages such as Python, or for a specific framework. For example, we have two examples here. They are very good approaches. They are very useful approaches, but they are fixed to specific languages such as NoWorkflow that works only with the Python scripts. He takes a Python script as input and collects all provenance data during the execution of this script. And Samba, that's focused on Spark application. So it receives the Spark application, Spark code as input, and then during execution, collect all information, all provenance information related to that execution. But most of the scripts are not Python. Many of the scripts are not Python or implemented in Apache Spark. So we need a solution that's agnostic of framework or programming language. So we proposed during the PhD time of Vitor Silva, that's another student, a tool named DF Analyzer. The idea of DF Analyzer is to provide an agnostic way for scientists to define what type of information he or she wants to capture. And then the DF Analyzer, it's a library that we couple to our code, to our own scripts. Based on instrumentation, we have to call the DF Analyzer in some part of the code. I will show how to do that. And then during the execution of the script, during the execution of our program, the DF Analyzer captures the information that we define that's important and stores in a database so we can query at runtime and we can do our analysis at runtime. DF Analyzer was published at VLDB. It's a major conference on database domain. And we have a journal paper on Software X that describes the tool and in very specific details. So once is interested in that, it's easy to achieve that. Sorry. So this is the original architecture of the DF Analyzer. We have four modules, four main modules, if you can say that. The first one is where we have the application. So we have to the code that we are going to execute and have to instruments to put some markups on the code to inform where the DF Analyzer needs to start to collect improvements or to register the execution. We have a storage module where we store the capture provenance data and the raw data files. So all raw data files produced by the script are stored in the storage area. And we store in the MonadDB, that's a columnar database, all the provenance data captured. In the capture module, we have the components that extract the information that the scientist informs that's important for her or for him. So, and we can also index the raw data. We did not index in the context of this paper, but there is a component that it's possible to do that. And we have a query interface and analysis module that provides a way for scientists to query the results without the need to access the MonadDB, that's a WFMS, to access the data. All the data collected by the DF Analyzer is stored in a database that's compliant to a W3C standard. That's a provenance standard of W3C. That's named PROV. This module capture all the specification of the script of the data flow. So Here we have the data flow that we are modeling, the activities of the data flow that's called data transformations. We know what parameters and data sets are used in a specific execution. So we collected all the prospective provenance, the structure of the experiment, and the retrospective provenance, that's the information about the execution of the experiment. It's all together in the same database, so the scientist is able to query that at runtime and after the execution, of course, as well, but mainly at the runtime to provide the data analysis. We evaluated this approach using a phylogenomic workflows that is already public and already available to use. That's named Phylogenomics. The idea of Phylogenomics is to producing phylogenomic trees based on an input data set of protein sequence of genomes and to infer evolutionary relationships among the organisms that were provided as input. Phylogenomics structure is presented here. We have first sub-workflow, that's a phylogenetic analysis workflow. It's composed by 4 activities. We perform first multiple sequence alignment, then format this sequence alignment, then we choose the evolutionary model and produce the phylogenetic tree. For each of these activities, we have 1 or more programs associated. After that, we perform a quality analysis to analyze the produced phylogenetic trees. We have a step of MSA concatenation, concatenate all the alignments produced in the first sub-workflow. So we have here a super alignment, and then repeat the activities, choosing the evolutionary model, and then constructing the phylogenomic tree. So this workflow is data and compute intensive. So depending on the amount of data that we provide as input, it can execute for hours or even days, depending on the machine and the volume of input data. But we use that, and then we use that as use case to evaluate the F analyzer in the context of bioinformatic domains. And we, of course, added new mechanisms for the F analyzer to achieve and to produce some interesting queries that the users want. So now I'm going to present how we can instrument the code to call the F analyzer. The first part is to insert a part where we define the prospective provenance, the structure of what we're going to capture. In this part of the code, the scientists will define what type of information is interesting to store at the provenance database. So here I defined the data flow. I'm going to define the start of the data flow. And here I'm going to define the transformations that are inside my code. So here I have a transformation named alignment model. I'm naming that first, and then I'm defining what type of input data it receives. And here we have a directory of input data, the alignment, and a sequence or a code, and produces as output an alignment, a new directory, and a code. So I'm defining what type of parameters it's important to capture of the input of this transformation and output of this transformation. So here the scientist defines what are the important information for him or for her. We do not capture all of the parameters involved in the script. So the scripts may have some 1,000 parameters. So, but in this case, we are going to capture just these 3 the input parameters and just these 3 input output parameters. After that, we associated, the data sets with the transformation, and that adds the transformation to the data flow. We have to repeat that for each data transformation that are identifying in our codes. For example, in the Phylogenomics, each of these boxes, we consider data transformation. So we have to program to instrument the codes to represent each of these data transformation within our code. And then when we achieve the part, when we find the part where the transformation is effectively executed in the code, we have to define some tasks. The tasks represent in our code whereas the code is effectively being executed. So this previous part, we can add in the beginning of the code. That's prospective provenance is like a template, what we're going to capture. But this second part here, we have to put exactly where the transformation is being executed. So I identify at the code, at the Python program or Perl program, for example, where the remove pipe model is being executed. Before that, I have to define the task, and then I define where this task begins. So when the F-analyzer is analyzing the input code, we identify, okay, we have to start capturing here. We process what the original script is doing. And after that, we start, okay, it's the end. We have to stop capturing information. And what we're going to capture, what the scientist defines. In this case, the directory parameter that was defined in the prospective provenance, we have to define what variables in our code are associated with that parameter. So in this case here, we are defining that, so the F-analyzer can capture. There's no miracle here. We have to associate the variables of our code to the parameters defined in the template. When you do that, the tool automatically creates the database. So for each one of the data sets defined in the prospective provenance, an input or output data set, we created a table in the database to collect all information. The user does not need to know anything about how to manage the database. So the tool automatically creates database and stores all data in this created database. So here we have the table tasks that represents all executions of data transformations within our code. And we have here the relations, the parameters that we are going to consume or produce for a specific task. For each one of these data sets that I specify in the code, I created a specific table in the provenance database. So a modification that we performed, that Gustavo performed in the code of the F-analyzer, is to add a telemetry data extractor. So besides collecting the provenance data and domain-specific data within parameters and files, we also collected telemetry data, so performance data associated to the execution of the script. We collected how much CPU we are using, how much memory we are consuming, how much disk we are consuming. So it's important sometimes for analyzing the performance of the script, the performance of the application, both for by informatician or the administrator or the distributed environment. It depends, but it's quite useful. Let's check some examples of queries. The first one, it's about domain-specific data. We capture the chosen evolutionary model for each one of the executions. We capture what was the chosen evolutionary model after the execution of mod generator application. So a simple query, you can analyze how many times each model, each evolutionary model was used in several executions. For example, in this case, the WAG model was the most used for the input data. So we can analyze that, or you can query for a specific execution. Here we are sum up, we're using the count, we are counting how many times each model was used, but we can, of course, check what was the chosen model for a specific input sequence or for a specific input data set. It's also possible. We just have not used the aggregation function here in the SQL code. Another analysis is about the execution time. Here we have the same workflow using Mr. Base for generating the tree and Haxmill for generating the tree. And here varying the type of the multiple sequence alignment programs. For example, using Mr. Base for generating tree and plus TLW for generating the alignment, we have a specific time. For using math to have it, so we can compare the performance of the multiple executions depending on the structure of the workflow, since we capture all this information automatically for the scientists. Another example is here. We have the average megabytes written by a specific execution. So here we have an execution using math as alignment program and Haxmill to produce the trees. So it's possible to check how many megabytes of data were written and how much megabytes of data were read. So this type of information is quite useful, and maybe it's captured automatically by the analyzer, this modification of the analyzer that we are providing for you. So as conclusion, the idea here is to provide a way, to provide a tool to capture provenance and to allow this runtime data analysis without the need to implement that or to control that. The only thing that the scientists need to do is to instrument the code, to define what type of information he or she wants to capture and where in the code he wants to capture that. DF Analyzer, it can be used in a single execution in our machine or in a HPC environments, in a cluster, in a cloud, because it's scalable. So in this case, we specialized the DF Analyzer for this phylogenomic experiment, and we added the telemetry capture, automatic telemetry capture, to provide performance metrics analysis at runtime as well. All the code of the DF Analyzer is available at GitHub, so here is the link. If you have some questions or doubts or suggestions, you can send an email. We are open to collaboration. We want users for our approach, so you are mostly welcome to use the DF Analyzer and to make some suggestions and to say if you found some bugs, find some bugs, but it's okay. We are open to collaborations. I'd like to thank the funding agencies, CNPq, FAPERJ, and CAPES, and thank you all of you for your attention in this presentation. Thank you very much.
Okay, Daniel, thank you very much for your nice presentation. I think that we have time, plenty of time for questions. I don't know, let's see in the chat. Not that much. Anyway, so I have one question, Daniel. It's really nice, really interesting, because we have been fighting in phylogenetics here in my team, and but again, the same as you put in your presentation. We are writing the scripts, so we have some part of the script written in Python, other parts written in R. So anyway, you know how this can happen. So we have mix of codes, and I did some, and the other colleague did the other part. So it's a mess. So the way that we find out, to organize this is to have some directory. So we have folders for each one of the projects, and then we organize in this way. But it would be really nice to have a database. You have somewhere to organize this, as you put, having this whole workflow. Easy to see and easy to reproduce. I think that's the main point here is the reproducibility. And I was wondering, for instance, do I have to do this? For instance, in my example, since I have this folder called project, for each one of these projects that in the end people should have some phylogenomic tree, we should have some pipelines. So we should have some workflow for each one of these projects. And for instance, if I understand correctly, I have to have a database for each one of these projects, or do I have to run this only once, for instance, for all folders, all projects?
Okay.
I don't know if
that's a very nice question. You can follow both approaches. We can create isolated databases for each one of the experiments. It's your option, but we can create a single database for all of the workflows, and within this database, you create different schemas for each one of your workflows. So they are separated within the database, the same database for management of the execution. But since they are within the same database, you can, for example, make some queries that associate data from different schemas. For example, I want to use the input of a workflow, that's the output of another. So you can perform a query that cross information between two schemas. It's possible. We did not do that in this experiment presented in this paper, but it's possible within the DF Analyzer.
And in the other way, for instance, let's say that I do this for the first time. So how can I use DF Analyzer? So in order to use DF Analyzer, I should have the code, all the pipeline, all the workflow, like say the code to use this, or can, let's say I'm just started now my project, and I say, let me say, okay, we did script for the alignment, but anyway, I first have to have the code, then I go to that analyze. So this is the path for the alignment, and I use the muscle, and I use the map to whatever, and then I go to the second part. So the point that I should have the code before.
Okay.
I should have this, or I can go directly for that analyze and trying to.
You can, for example, if you have the code, if you program the code, it's okay. It's how I present in the presentation. You have to instrument, you have to mark up on your code. It's okay. If you are using black box application, for example, math, you do not have the code of math. So I'm invoking the math within my Python script, for example. Okay. You can create a task in the F analyzer. So before the execution math, before invoking math in your code, you are going to start this task. And after executing math, you are going to stop this task. So we are marking where in the code math is being executed. We cannot instrument the math code, but we can, within our script, open the file that math produces and import to the F analyzer the content of this file. It's possible to do. So if some parameters is within the .math file, we can import that to the database, so we can use that content in your queries, in your analysis. It's possible to do. You cannot instrument the math code, but within your Python script, you can define where the math starts to execute and when it finishes the execution, and then we can import, you can extract information within the produced files. It's possible.
Okay, that's nice. So there's lots of flexibility, I mean, in order to use MDF analyze. And the last question, I think it's more like, for probably there are other colleagues, that they use, Galaxy, Galaxy workflow. And how can you integrate MDF analyze with Galaxy, or do they do the same thing? For instance, I already have some workflow, let's say, in Galaxy for the phylogenomic analysis, let's say standard offline, and I have now the analyze, so I can work with all together. They are competitors. So what?
They are not really competitors, because the idea of Galaxy and others workflow systems is to manage the execution. So we define the pipeline and Galaxy execute distributed in the cloud. That's not the idea of the F analysis. The F analysis does not control the execution of the application. We only capture information to provide the data analysis. But in this sense, Galaxy also collects provenance of the pipeline.
Yeah.
So what we did in a different experiment is to try to associate data from the F analyzer with the existing workflow system. We are still working on that, but we have a system that our group implemented that's named Cycumulus. So we collected part of the provenance in Cycumulus and try to associate that what the F analyzer captures. So in this sense, we could associate the data that Galaxy already captures with the data that the F analyzer is capturing.
Okay, that's really nice. I think that's the great contribution. That's the idea of have all these in one place, and that allowing everyone to indeed to reproduce. You can reproduce all the steps, and you can reproduce all these projects. And I think that for everyone that works in bioinformatics know that really hard, and sometimes it's really difficult. To reproduce. I think that's really nice, and that we have this your presentation in the end, because I think that the whole section. I don't know if you have any other question, but I think that if you don't have any other question, I think that we can close the session right now. And I'd like to say that it was I was honored. For chairing this session. It was really many interesting papers presented here, and all the papers in the beginning of the presentation. They present more about machine learning, how can people find data sets, and that's the idea. Because you cannot have a good model if you don't have a good data. And now you are bringing all the points. All the questions in the pipeline that's about reproducibility, because that's one of the questions that I made for one of the first presenters. That how can you manage to run again all these models, and how long does it take to run all these models? This kind of question that's really tricky, and it's really hard. For everyone that's listening now here and attended these, I think that most of people have already fight in trying to reproduce some paper that they found out, and then they see that in the end, sometimes the guys, they don't provide all the information, and sometimes they don't have the Git for, for the project. And I think that reproducibility is part of the question. And it's really interesting. Thank you very much, Daniel, for your presentation. And I hope that we can try to analyze. We should keep in contact, because it is for real. We have many problems that we have here in the ITV. We deal with phylogenomics and evolution. And my question was really, tricky, because we have been studying lots of cases now, and we have, for instance, we do the phylogenomics for aquatic species, and then we go for all the microbiota, and then we go for invertebrates, and there are different stuff. So that's what I said. That's a different project, but it's really interesting that we can have, some way, idea of building up this to help reproducibility. Okay, so thank you very much, Daniel. Thank you all the guys that stay logged in for the BSB session, and hope that everyone enjoyed this session today, and invite you all for tomorrow, so that we continue the conference. Thank you very much. And Daniel, I don't know if you that we closed all the stuff, but
yeah, I'm stopping live streaming to YouTube. So I'd like to remember that you have a morning session tomorrow, 9:30 a.m. So I'd like to ask the authors to arrive 30 minutes earlier, as we did today, to test the screen sharing. It's okay. So thank you for attending this session. I'd like to ask everybody that's here to open the camera so we can capture a picture of the participants, if it's possible. Just a moment. One, two, three. Thank you very much, people.
Thank you very much. Have a nice week and